Hello, in this video we will discuss about the endoplasmic reticulum and the damage of the endoplasmic reticulum to produce the reactive oxygen species ROS, the reactive oxygen species. So what is the role of endoplasmic reticulum? Endoplasmic reticulum is used for the detoxification of drug present in the when liver. On the other hand, it is the maturation of the protein and the conjugated protein also. So this is a cell which that is contain mitochondria, endoplasmic reticulum, cytoplasm and plasma membrane as well as nucleus. So this is the normal structure of the cell. So endoplasmic reticulum is highly sensitive to redox status and altered. The reactive oxygen species signaling can influence the protein folding and calcium release and mitochondrial respiration. So the mitochondrial respiration, this is very important. And in this way, the endoplasmic reticulum is highly sensitive to redox status. So it means the reduction. So in this way here I am drawing the all cell structure which that contain a plasma membrane and cytoplasm as well as the endoplasmic reticulum and mitochondria. This is the cell membrane. And this is the cytoplasm. Cytoplasm contain a cytosol. While this is the endoplasmic reticulum. This endoplasmic reticulum is basically organelle, is the single membrane organelle contain calcium inside and also used for the detoxification of drugs as well as the synthesis of protein, the maturation of the protein and the ribosome also present, and the below the endoplasmic reticulum the mitochondria I draw. So this is the trimeric complex protein is known as the ATF6, PERK and IRE1 alpha. So what is the IRE1 alpha? Is the inositol requiring enzyme 1 alpha. So this is the NOx4, the NADPH oxidase enzyme 4, which that oxidize the NADPH and into the NAD plus and also produce the reactive oxygen species when the hypoxia will occur. So when the decrease of the oxygen will lead to produce the more reactive oxygen species. So remember, and these free radical containing reactive oxygen species will react with the plasma membrane, the membrane of the endoplasmic reticulum as well as the mitochondria. So in this way, here also the plasma membrane contain a NOx2, the NADH oxidase enzyme 2, and in this way here you can see the NOx2. This basically is the calmodulin dependent kinase 2. Calmodulin dependent kinase 2 enzyme basically is NADPH oxidase, and in this way will produce more reactive oxygen species when hypoxia will occur. So this is the trimeric protein, the ATF6, PERK, and the IRE1 alpha, which that is bound with the dimerically inside that you can understand. So in this way you can understand this is the GRP 78 protein, while the here is the ERO1 is mean the endoplasmic reticulum oxyreductine. Oxyreductine means the oxidation and reduction also occur both and this protein. For example, and IRE1 alpha is used for the inositol requiring enzyme 1 alpha. So this trimeric protein will trigger due to the an unfolded protein response. Unfolded protein response will basically after this will trigger the ERO1, the endoplasmic reticulum oxyreductine protein activation, and in this way it will trigger the to use the oxygen to convert the hydrogen peroxide. to use the oxygen to convert the hydrogen peroxide. So remember, and in this way this ERO1, which that again to convert the ERO1, and in this way here you can see the oxygen will convert into the hydrogen peroxide, and on the other hand, so remember the glutathione, which that will convert into the glutathione disulfide, so the two sulfide bond which that will be occur, and in this way glutathione peroxidase enzyme, the glutathione peroxidase enzyme 78, which that will get the reactive oxygen species scavenger, which that is known as the antioxidant, will work to produce the water from the hydrogen peroxide to decrease the reactive oxygen species. So that means it is the antioxidant and reactive oxygen species scavenger, so scavenging of the reactive oxygen species, and in this way this reactive oxygen species scavenger, the glutathione peroxidase, will convert the inactivation the GRP78 into the GRP78. Again, this GRP78 is known as the glucose regulated protein activation. So this is also bind with dimerically with the trimeric protein, the ATF6 and PERK and IRE1 alpha. So on the other hand, here you can see the basically here is the peroxyredoxin, which that will be convert the peroxyredoxin 4, peroxyredoxin 4, which that will be activate, and after this activation we will learn. So here is the reactive oxygen species when enhanced and increased will lead to oxidation of phospholipid bilayer of plasma membrane or the endoplasmic reticulum membrane or mitochondrial membrane. So on the other hand, here is the protein disulfide isomerase enzyme 1. This protein disulfide isomerase enzyme basically used to convert the protein to modified by the disulfide bond. So that is disulfide bond isomerization, not a modification, but it is change the location for changing of the shape of the protein. So that is why the modifying the protein into the endoplasmic reticulum. So that is known as the maturation of protein. While on the other hand, the protein disulfide isomerase will also peroxyredoxin will use to produce this protein, and in this case you should need to understand when the reactive oxygen species increase will lead to misfolding. Why misfolding? Because the reactive oxygen species will react with the protein to addition of the electron will change the morphology of the protein and their structure. So increase the chaperone activity will increase. So the chaperone activity will increase due to the glucose regulated protein activity. While here is you can see the endoplasmic reticulum oxidoreductin 1 enzyme, which that will lead to calcium outflux due to the inositol 1,4,5 phosphate receptor, which that will require the IP3 binding this receptor. After this binding, the efflux of the calcium is very important. So in this way the efflux of the calcium will occur. So when efflux of the calcium will occur, which that go into the mitochondria. So after this calcium, from the mitochondrial calcium uniport, this mitochondrial calcium uniport channel, which that required the inner mitochondrial membrane to influx of the calcium. So that is why the inner mitochondrial matrix will increase the calcium. So calcium will increase will lead to use the respiration, lead to increase the reactive oxygen species. While on the other hand, here is the calcium called modulin dependent kinase 2 will regulate bind with the calcium, will trigger more NADPH oxidase, will increase the reactive oxygen species in this manner. So on the other hand, the calcium will increase in the inner mitochondrial matrix. For example, the mitochondria is also store the calcium, but increase of the calcium will lead to reactive oxygen species increase. While this is the electron transport chain run in the inner mitochondrial membrane, so the NAD plus after the reduction will lead to oxidation. Will lead to outflux of the proton, while the electron will be accept to transfer the quinone to ubiquinone, and in this way the complex 2 and complex 3 which that will release the reactive oxygen species, and in this way the cytochrome which that will electron used to produce the protein 66HSHC, and in this way the oxygen which that will be triggered to produce hydrogen peroxide through protein 66, and this hydrogen peroxide will go into the cytoplasm, and here is the proton will influx will lead to produce ATP. So remember, so in this way here you can see the reactive oxygen species will move out from the mitochondria, which that will lead to P66 protein SHC, which that will lead to intercellular and intracellular stress, will lead to reactive oxygen species enhance. So in this way, the mitochondria will produce reactive oxygen species due to the more decreasing of the oxygen. So the hypoxia will lead to, and there is another way, and also the reactive oxygen species will lead to inflammation, to immune cell activation. We will discuss next.